months now. And it's a student-led initiative where we invite speakers from all over the world to come share with us their stories. And today we have with us Dr. Sheila Teves, and I would like to invite my colleague Sonu to introduce her.
Hello everyone, Dr. Sheila Teves is here for today's CCMB Biologue, and she is an assistant professor at the University of British Columbia in Department of Biochemistry and Molecular Biology. She did her PhD with Dr. Stephen Henikoff in University of Washington, and there she studies about transcription dynamics and how nucleosome positioning affect transcription. She did her postdoc with Dr. Robert Tejan at the University of California, Berkeley. So there she studied transcription status of particular, how this transcription status is maintained during cell division. Now her lab is interested in working on transcription memory, like after each cell division, how the cell remembers which set of genes to be turned on and which are to be turned off. So over to you, Dr. Sheila.
Thank you so much for the invitation and the introduction. I'm going to start sharing my screen. Okay, so so today I'm going to talk about projects in the lab dealing with transcriptional memory and plasticity in embryonic stem cells. Just a quick land acknowledgement. I'm joining you today from UBC, which is located on the traditional, ancestral, and unceded territory of the Musqueam people. And we are very lucky to be able to live, play, and work in such a beautiful area. So my lab studies transcription regulation, as you might have guessed from the title. But why do we study transcription? Well, I've always been fascinated by the fact that we all start from a single cell, the zygote, which develops into a fetus and then hopefully to a highly functioning adult. And so what this means is that we all come all the cells in our body share a single genome for the most part. And yet we have these very diverse cell types in the body that function with very specific function. And so how do we go from one genome to many different cell types? And so this is a very important part of development, and it's why I'm most interested in transcription regulation. But transcription is very important for many other things, such as I just mentioned, development, but also response to stimuli. And right now we have a lot of ongoing research with a very big response to stimuli, which is infection to SARS-CoV-2. And other things like circadian rhythm, how our body has its own unique rhythm that is on a 24-hour cycle, but then each cell within the body also has its own internal clock. That's all regulated by transcription. But of course, another aspect of transcription is that any type of, a big type of research that's devoted to it is when transcription goes wrong. And so diseases like cancer, for example, is a major component of diseases like cancer is errors in transcription regulation. And so these are the motivations that we have for studying transcription. In my lab, we use embryonic stem cells as a model system, primarily because they have two very important properties. They can self-maintain and divide and form new embryonic stem cells, but they also have the potential to differentiate to many different cell types of the body. And so these processes are really a great system for studying transcriptional memory for when cells maintain their identity, but also transcriptional plasticity for when they want to change their identity to a new cell type. And so these are the kinds of questions that we're interested in. And we study transcription regulation using two very different techniques, actually. We use genomics to look at large-scale genome-wide data. Basically, we want to know where in the genome DNA binding proteins bind, or which genes are turned on and which ones aren't, these types of questions. But also, we, on the flip side, we use microscopy and specifically live cell single molecule imaging to look at the dynamics of these processes. And here we zoom in at a single factor at a time, but the resolution and temporal dynamics that we get is incomparable to what we would have with the genomics data. And together, combined, we get really comprehensive information about the general processes that we're studying. So we really try to harness these two different techniques. And so today I wanted to talk to you guys about projects in the lab that are primarily dealing with transcriptional memory side of our research. So the first part of the talk, I'll be talking about our projects on mitotic bookmarking, led by postdoctoral fellow Merrick Budzinski and Rachel Price, graduate student Rachel Price. And then the latter half of the talk, I'll focus on mechanisms of transcription. So we'll try to get into the nitty-gritty details of how transcription works. And this project is led by two graduate students, James Kwan and Thomas Nguyen. There's a third project in the lab going on that I won't be talking about today, but we're really excited about it because it's a new direction for our group. And it's on the evolution of transcription mechanisms. Basically, how does something so highly conserved, transcription going from DNA to RNA, which all living organisms do, how do you maintain that conservation while diversifying to allow for very diverse organisms? But anyway, hopefully in the near future, you guys will invite me again and I can talk about that project as it develops. But for now, I'll focus on the first part, which is on mitotic bookmarking. Okay, so most people don't realize, but the cell cycle is actually highly disruptive to transcription. So as cells go from G1 and then they grow and then they replicate their genome, DNA replication is mutually exclusive to transcription. And then as they grow some more in G2 and start to enter into mitosis, there's these massive changes in cellular organization. So as you might see here, the green marks the chromosomes, and you can see massive changes. You have chromosomes condensing into mitotic chromosomes. You have the disassembly of the nuclear membrane and major reorganization in the cytoplasm. And then also you have the global downregulation or inactivation of transcription happening during mitosis. And so one of the things that I've always been interested in is how is transcription maintained after mitosis? That is, once the cells divide, how do the new daughter cells know which genes to turn back on and which ones to keep silenced? And so with this question in mind, one of the major theories that's been proposed to maintain transcription programs through mitosis has been termed mitotic bookmarking. And that really highlights the role of transcription factors in this process. Well, transcription factors are really important for regulating transcription. They are DNA binding proteins that bind to specific sites in the genome and then recruit the general machinery to activate transcription. We can generally measure where transcription factors are bound throughout the genome by the accessibility of chromatin. And here, accessibility just means that it's accessible. And the higher the peak is, the more accessible the region is. And it's usually enriched for transcription factor binding sites. And so what does this mean for mitosis? Well, previous studies have shown that transcription factors appeared to be largely excluded from mitotic chromosomes. So here's an immunofluorescence immunofluorescence of a transcription factor YY1 in red, showing negative staining throughout mitosis relative to DNA, which is stained here in DAPI in blue. And this is just for one transcription factor, but it's been shown over the past few decades for many, many different transcription factors. And so even though it wasn't quite clear then, like how would transcription factors promote mitotic or transcriptional memory through mitosis? Well, it was then a big advance that certain transcription factors were identified to remain bound to mitotic chromosomes. So here's FOXA1, a transcription factor important in liver tissues. And under live cell condition, they see it highly enriched on mitotic chromosomes. And so it was around this time when I thought, could this mechanism be used to maintain ESL transcription program? And so to study this, I focused on a pretty key transcription factor for ESLs, and that's SOX2. And one thing that was, I'm breezing through this data because it's been published a few years now, but one of the main findings that we found is that SOX2 under live condition is highly enriched on mitotic chromosomes, as you can see here. So this is a live cell picture. But the weird thing that we found is that once we add formaldehyde, which I'll show you in the movie here, it starts to get evicted. So in 10 seconds, formaldehyde will be added, and then you can see under live conditions, transcription factors are now being evicted from mitotic chromosomes. I'll just show that again. And so this really led us to this discovery that perhaps even though people have seen transcription factors excluded, perhaps it's not really the real biology, and it's rather an artifact of the fixation. And in fact, most of the experiments that have shown this have been using immunofluorescence. And one of the first things you need to do for immunofluorescence is fix your samples. And so this is exactly what I just said. The evidence for transcription factor exclusion during mitosis is largely from immunofluorescence. And this is problematic because the first step of immunofluorescence is formaldehyde fixation. And so what we found is that SOX2 and many other transcription factors actually interact with mitotic chromosomes, in contrast to decades worth of literature. And so the next thing we wanted to ask is, how does SOX2 interact with mitotic chromosomes? And is it different with the way the transcription factor interacts with interface chromatin when it's not condensed? And so we specifically asked two questions. What fraction of SOX2 is bound, meaning how much of the protein is bound within any given time? And what is the dynamics of the binding? Like, how is it finding its DNA target sites? And so to really get at these questions, we really have to use imaging technology. And this is where a single molecule live cell imaging really shines. So in our lab, we use single molecule imaging in two different ways. First is the fast tracking. Here we're taking 200 pictures per second, about 200 pictures per second, and we can identify the fast diffusion properties of each molecule. So here, each individual spot that you see is a single SOX2 molecule that's diffusing pretty rapidly throughout the nucleus. And using this technique, we can actually calculate how much of the protein is bound versus freely diffusing, and we can assign diffusion constants for each population. A second way of doing single molecule imaging is called slow tracking. Here, instead of 200 frames per second, now we slow it down and we take 2 pictures per second, so 2 orders of magnitude slower. And what this does is that when you keep the camera open in this slower time frame, any fast-moving molecule that's moving in front of the camera will get blurred out, in the same way that if you take your camera phone and you move in front of it while taking a picture, you'll look blurred. And so all the blurriness over here, the haze, are all the individual molecules that are moving way too fast for the camera. Instead, what this does is it isolates only the bound molecules. And so then what you're left with are individual SOX2 molecules binding to their DNA target sites. And using this, we can identify specific versus non-specific binding, and we can assign residence times for each population, meaning how long do they bind the DNA. And so I'm just gonna show you how we looked at SOX2 binding to mitotic chromosomes using flow tracking. As I mentioned, this means that we're taking 2 pictures per second, so 500 millisecond frame rates. This leads to blurring out of fast diffusing molecules. And then we can measure the length of time the molecules bind to a DNA by just counting the number of frames. There's a lot of mathematical modeling involved in this. And so we can present the data in this way, meaning 100% of the molecules are calculated as bound at time 0. And then over time, we calculate how much is remaining bound. So this is called a decay curve. And so in gray here shows the decay curve for SOX2 in chromatin, whereas in mitosis is in red. And qualitatively, you can see that the plots are different. But quantitatively, what this means is that SOX2 during mitosis is binding on and off faster than it's doing so during interphase. In fact, we can actually quantify this using mathematical modeling, and we can assign residence time for SOX2 in interphase, which is we on average we see about 12 seconds DNA binding times, and then in mitosis, this residence time is decreased by half. And so what that means is that every time it comes on the DNA, it comes off the DNA twice as fast as it does on interphase chromatin. And so SOX2 binding during mitosis is more dynamic than it is in interphase. And so why would this be dynamic? We tried to answer that using different ways, including we wanted to ask where in the genome SOX2 is binding during mitosis. And so to do this, normally people use a very standard protocol called CHIP, chromatin immunoprecipitation, where you cross-link your DNA, you shear the DNA fragments, and then you pull down with specific antibody and then molecular techniques to lead to sequencing. But I just told you that cross-linking is a very problematic thing for our purposes. And so we wanted to avoid this, this method. And so we turned to cut and tag, which is a new method where it's under native conditions. The cells are instead permeabilized, and then that allows for the diffusion of specific antibodies into the nucleus, where it can then bind to its target protein, and then introduce this protein A TN5 fusion, such that the protein A will get attracted to the antibody, and the TN5 will integrate sequencing adapters into the neighboring DNA sequences, and then you can just amplify your library and go directly to sequencing. And what this really allows us is to probe the DNA binding properties of transcription factors under native conditions. So that's a big highlight for us. Okay, so we performed cut and tag for SOX2 in asynchronous population and in mitotically synchronized populations. And I'm showing you here a specific loci that SOX2 binds in asynchronous. And in the first part here is a region where SOX2 is bound in asynchronous, and it's also bound during mitosis. But not all SOX2 binding sites are bound during mitosis. For example, here's one where it's bound during asynchronous population, but in mitosis, its binding is greatly reduced. But overall, the binding of SOX2 in mitosis is very reproducible of asynchronous population. And so for the most part, where SOX2 binds in chromatin, interface chromatin, are the same locations to where it binds in mitosis when the chromosomes are highly condensed. So we now know that SOX2 binds site-specifically during mitosis. And then we're starting to question, like, what are the molecular determinants of SOX2 bookmarking? What makes SOX2 able to bind to mitotic chromosomes? And to do this, we actually look at pairs of transcription factors, meaning they share the same DNA binding domain. But what's interesting is that they have very different mitotic binding capacity. So for example, SOX2 here is enriched on mitotic chromosomes, but a very similar protein in the same family, SOX13, which share 55% similarity, is actually excluded from mitotic chromosomes. In parallel, we're also exploring another pair of transcription factors, HSF1 and HSF2, that also have the same type of mitotic DNA binding. And so what we were asking is, what is the role of the DNA binding domain in mitotic bookmarking? And one of the first things we did was to swap the DNA binding domain between these pairs of very similar transcription factors. And then we asked, how does that change the behavior of mitotic binding? And so here, as I just showed you, SOX2 on mitotic chromosomes, highly enriched. And the chromosomes here are marked by H2B GFP. In contrast, the very similar SOX13 is excluded from mitotic chromosomes, you can see here. Now, when we swap the SOX2 DBD with the SOX13 DNA binding domain, now we see that SOX2 protein with the SOX13 DBD is now excluded from mitotic chromosomes. So it's lost the capacity to bind to mitotic DNA. Whereas in the converse, SOX13 with the SOX2 DBD has now gained the capacity to bind to mitotic chromosomes. So we think that there is something in the DNA binding domain that is sufficient to confer chromosome binding. And so where this is going now, we're looking at whether SOX2 DBD is sufficient for specific binding. So I'm showing you here overall association with mitotic chromosomes, but whether these swaps are actually binding to their specific target sites, that is another question that we're trying to answer. And so where that is going now, we're looking at applying, again, cotton tag to our transcription factors and their respective swaps, and then applying single molecule imaging to our transcription factors and their swaps to see how their behaviors in mitotic chromosomes are now maintained or altered depending on their DNA binding domain content. Okay, so that is kind of where we are with the mitotic bookmarking side of the lab. So I'll take this moment to switch gears and talk about still on transcription, but a very different type of project, and this time looking at the mechanisms of transcription. So I've been focusing a lot on transcription factors, which are largely working at enhancers or proximal promoter regions, but eventually all transcription goes down to the preinitiation complex, which is the RNA polymerase II complex that binds to the core promoter region and actually transcribes the DNA. And the assembly of this giant complex is highly regulated and very stepwise. And it involves a very large multi-subunit different groups of proteins. At the center of this is the first protein that's supposed to bind to DNA, which is TF2D with the TBP subunit. And TBP is a really interesting protein because it's actually required for all 3 RNA polymerases. So here are the 3 polymerases and their specific structures. And TBP is highlighted here in pink. I think it's pink. And you can see that its location and general overall structure is very conserved across the 3 polymerases. So TBP is at the heart of all 3 structures. In fact, TBP is a highly, highly conserved transcription factor. It's found not only throughout all of the eukarya, but also throughout all of the eukarya, but also throughout archaea. And so here's an alignment of TBP and its core domain, core DNA binding domain, highlighted in gray, showing very, very high conservation across very diverse species. And it's this type of conservation, but also a specific region on the N-terminal domain that is divergent, and it seems to have species-specific function. And so this is the project I was alluding to earlier, where we're looking at the evolution of transcription mechanisms. But back to where we are in transcription mechanisms, from also, again, decades work of pioneering research, right? We've learned from back then, 1980s, that TBP is essential for transcription in vitro and in vivo. So here is purified yeast proteins, and whether they transcribe and generate RNA in vitro is dependent on the presence of the yeast TBP protein. Okay, so it's essential. And here's a much more recent study back in 2019, also in yeast, where they depleted individual members of the Preinitiation complex, including TBP here, and then they looked at Pol2 binding at this gene, but also genome-wide. And what they showed is that depletion of each member, specifically TBP, resulted in a very decreased Pol2 binding and Pol2 activity. So TBP is essential for transcription. But what's interesting is that there are some incidences where this doesn't seem to be the case. So here's a study back in 2002 where they were looking at mouse embryos where they knocked out TBP. These embryos eventually die because TBP is essential. But at this stage, they were able to isolate blastocysts that are still alive, but they don't have any TBP. So here they showed no TBP by immunofluorescence to the anti-TBP, and here are all the DNA. But what's interesting is that at this stage, there's actually transcription, as evidenced by incorporation of bromouridine RNA. And so how is there transcription without TBP? It wasn't really quite clear. This was quite a short paper, and it was, as far as we know, wasn't really followed up. And so we got into this very fortuitously because we had generated an auxin-inducible degron fused TBP at the endogenous locus. And so we are able to use the drug auxin to rapidly deplete TBP using the ubiquitin mediated degradation pathway. So within 6 hours, we're able to see over 90% degradation of TBP, which is the endogenous TBP is unaffected. Sorry, wild type TBP. These are all knock-ins, so all endogenous proteins are the AID TBP. And so we wanted to make sure that TBP is depleted at all its binding sites. And so we used cut and tag again to profile TBP before and after TBP degradation. So here are the control conditions, TBP binding to the promoter regions of these genes, GAPDH and beta-actin. And then upon TBP depletion with IAA, you see it's mostly gone. And this is true genome-wide. I'll just start with the heat map here on the control. So all genes aligned at the transcription start site, and then plus or minus 1 kb upstream and downstream. And what you can see is that control conditions, TBP is really binding at the transcription start site. And then once it's depleted, it's mostly gone. And then the plots up here are the global averages of the heat maps. So again, blue in control, enriched at the transcription start site, and then depleted heavily when treated with IA. So we're fairly certain that over 90% reduction in TBP with auxin treatment. So what happens to transcription? Well, we looked at transcription in a couple of different ways. We profiled Pol2 binding with cut and tag, and then we measured nascent RNA production using a technique called NetSeq. I'm not really going to go over what NetSeq is, but just know that it's measuring newly transcribed RNA. And we're specifically interested in the newly transcribed RNA because we're treating these cells for 6 hours. And at that point, at that short time point, the global RNA levels aren't really changing. So we need to look at newly transcribed RNA. Okay, so what happens? So let's just look at, again, our favorite genes here, GAPDH and actin B. Again, TBP in orange here, before and after IA treatment, showing good depletion. Pol2 in green, control and IAA, and then newly transcribed RNA in blue. And what you can see in this in these results is that TBP depletion doesn't seem to affect Pol2 binding or its nascent RNA production, which is really weird for us. In fact, this is true globally. So here again is our TBP depletion genome-wide. And now when we look at Pol2 binding or new RNA, we see largely no effect on transcription. So again, here are all genes lined at the transcription start site with genes arranged by gene expression, decreasing gene expression. And you can see that there's really very little difference between the control and the TBP depleted conditions. And the same is true for the newly transcribed RNA. And so we can look at this in a different way. And here we have now taken it on a gene by gene basis, where each dot here is a gene in the mouse genome. And we're plotting on its y-axis its log-fold change between control and IAA. And on the x-axis, its average gene expression. So the higher expressing genes are on the right side, lower expressing genes on the left side. And basically, anything above the y-axis here would be upregulated, and anything below the y-axis here is downregulated by TBP depletion. And what I hope you can see is that what we would have expected, which is global downregulation, is actually not happening. Instead, what we see is only a handful of genes that are affected. So this is true for the Pol2 binding, and it's also the same for the NetSeq results. We only identify a handful of genes that are significantly downregulated. And here's a couple of these genes. This one is RAB26, and this one is EIF4A2. And you can see, yeah, it's actually downregulated with Pol2 binding and with nascent RNA, the same over here. But this is definitely the exception and not the rule. So we started to ask, well, do you need TBP when inducing transcription? Because one possibility is that these are steady state transcription levels. Maybe once it's established, you don't really need TBP anymore. So we thought, okay, can we induce transcription to go from 0 to active? And do you need TBP then? Well, one of the ways that we induce transcription is the heat shock response. So the heat shock response is a highly conserved defense mechanism against a lot of different stresses. And it involves the activation of transcription factors upon stress, which then bind to their target sites and activation of target genes. So these are called the heat shock protein genes. And another consequence of this is that you have a global downregulation of transcription. And the nice part about it is that it's reversible. When you remove the stress, the cells recover, and then during recovery, you have the global restart of transcription and then silencing of the heat shock protein genes. So we use this system to ask, is TBP needed to induce heat shock genes? And here's our experimental scheme. We either have cells growing with or without TBP, and then we heat shock the cells at 42 degrees for 30 minutes, and then we allow them to recover. And at this point, we collect the control cells and we collect the heat shock cells and the heat shock recovery cells. And then we perform Pol2 cut and tag and then net-seq. And I'm just going to show you here the control, the heat shock, and the heat shock when TBP is depleted. And so here, as you can see with TBP, for these two heat shock protein genes, that TBP is depleted upon IAA treatment, and that under normal conditions, these genes are off, and then upon heat shock, they're massively upregulated. But then what you can see here is that for these genes, without TBP, they're still massively upregulated. And then the same is true for the nascent RNA results. So what does this look like genome-wide? Again, here is the MA plot where each gene is a single dot, and anything that's red is significantly upregulated in the heat shock compared to control conditions, and anything that's blue is significantly downregulated in the heat shock versus control conditions. And when we compare the heat shock with TBP depletion versus the heat shock, you see no difference in gene regulation, suggesting that TBP depletion does not affect the induction of these genes. Okay, so we thought, okay, maybe we got unlucky and we chose a way of inducing genes that doesn't require TBP. So we thought we'd try something else. So these are embryonic stem cells. And so we thought, okay, let's try to differentiate them. And so we asked, is TBP needed to induce differentiation genes? In this stage, these are regulated very differently than heat shock. And we're treating them over 16 hours with or without TBP depletion, and then with or without retinoic acid differentiation. And again, here is the control where we show TBP is depleted in this differentiation gene. And then upon differentiation, we see upregulation of Pol2 on increased binding of Pol2 on this gene, and that this binding occurs even without TBP depletion, or even without TBP. And again, this is a global look where we see a lot of genes upregulated and downregulated in differentiated versus control cells. But when we look at the differentiated with TBP depletion versus just differentiated cells, we see no difference in the gene expression patterns. And so genes that are upregulated are still being upregulated even without TBP. So this really gets us to question, like, how is the preinitiation complex formation affected without TBP? Again, I'll remind you that TBP is part of the TF2D complex, which is one of the first factors to bind DNA and recruit the rest of the machinery. What that actually looks like is here on the cryo-EM structure of TF2D. This is the whole TF2D complex with the different subunits, TBP here in red, and here are all the different subunits of TF2D. And so what we did is to IP with against TAF4, pull down the TF2D complex, and then measure by mass spec in cells under normal conditions or with TBP depletion. And so I'm just going to orient you what the mass spec data looks like. So again, here are the volcano plots where we look at TAF4 IP versus IgG. So these are all significantly enriched in the IP compared to the IgG. So these are all the TAF proteins. You can see them all here, the TAFs, the TBP, and then etc. So we pull down the TF2D complex by using TAF4. So what happens when we do TAF4 IP in TBP depleted cells? Well, we can still pull down all the different subunits of TF2D. So you can see here the TAFs are here, TAF8, TAF9, etc. They're all there. And in fact, when we now compare the TAF4 IP under control versus TBP depletion, this quadrant here, which is where all the significantly enriched proteins are located, is now empty, meaning that there is really no difference between the complex that's pulled down either with or without TBP. So TF2D forms even in the absence of TBP. So what is TBP actually needed for? We've been kind of scratching our heads here. We thought that these should all have affected global POL2 transcription, but instead seems to have no effect with POL2 transcription. But I'll remind you that TBP is supposed to be required for all 3 RNA polymerases. And so we next looked at what happens to POL1 and or POL3 when TBP is depleted. And I'll just show you here what we looked at for POL3. So POL3 transcribes the tRNA genes. So here are several tRNA genes where we looked at TBP, again, under control and then depleted conditions. And then we measured RNA polymerase 3, and we can see that it binds to the tRNA gene. But then once we deplete TBP, POL3 is mostly gone. And that is true over here as well. This is true for all tRNA genes. So here are all tRNA genes lined by their transcription start site. These are really small genes, so it's very narrow. But you can see that TBP binds there under normal conditions and is depleted when TBP is gone. And then what happens to POL3? Unlike POL2, POL3 binding is severely affected by TBP depletion. So you can see here under control, POL3 is binding to the tRNA genes, but when we deplete cells with TBP, it's now mostly gone. And so what I've shown you on this part is that global POL2 transcription is mostly unaffected by TBP depletion. Gene induction is unaffected and does not require TBP either by heat shock or by differentiation. The TF2D complex formation is also not affected affected by TBP depletion. But instead, what we see is that TBP is required for Pol III transcription. And so we're following this up now. We're trying to get more at the mechanism of the precise role of TBP in these processes. And so that project has been summarized in a bioRxiv preprint and hopefully will be published soon. And so that's kind of all I have for you guys. I've given you 2 stories, one on mitotic bookmarking and another on mechanisms of transcription. And hopefully this gives you an idea of how we tackle questions in transcription regulation in embryonic stem cells. And with that, I will thank the lab, which did most of the work. I've highlighted the students and the postdoc that have done the work along the way. Our collaborators for the IPMS, Philip Lang and group. We are supported by grants listed down here, and I'm happy to take any questions.
That was very nice talk, Dr. Sheila. So now we'll take questions from Q&A. And I request audience, if you have any questions, you can raise your hand and then we'll unmute you and you can ask. So the first question is Pol1 transcription factor affected in TBP downregulation?
That's a great question, and it's actually the next direction that we're trying to look at now. We don't know. Pol1 transcription is more challenging to look at because of how repetitive the ribosomal DNA genes are and how abundant the ribosomal RNAs are. And so these things are a challenge for sequencing-based methods. And so we're trying to figure out ways to do exactly this question.
Okay. The next question is, why do transcription factor gets excluded from chromatin on fixation? Could this happen with other chromatin binding factors as well?
Yeah, that's a great question. I showed you guys one movie, but actually that one movie, it took about 2 years for us to get to that point of actually figuring out what is happening, because we kept getting one answer when we look at cells under live conditions, and then another answer when we look at them under fixed conditions. So we finally put it together after 2 years of of bashing our heads. But what we think is happening is, and we have a lot of data supporting this. So this is probably the easiest way to answer integrating all the data that we have. And so what we think is happening is that as formaldehyde diffuses into the cells, it goes from the outside in, right? So it has to diffuse from outside in. But as it's going from outside in, it cross-links as it goes, right? So anything that's unbound in the periphery gets cross-linked first. And then the ones, at least during mitosis, the factors that are bound at the center where the mitotic chromosomes are free to bind and unbind until formaldehyde finally gets there. But the end result of this is that you essentially have a depletion of unbound molecules, effective depletion, right? Because you cross-link them, so they can't come back to bind to mitotic chromosomes. Whereas the ones that are there at the center can still unbind. And so you then get this kind of reverse depletion that is dependent on the diffusion dynamics or kinetics of formaldehyde. And so what we found is that when you have DNA binding proteins that are really stably bound, so for a much longer time, they remain bound to mitotic chromosomes even after fixation. And so it seems to be like a race. How fast can formaldehyde get there and fix you without you coming off, unbinding and binding again. And so that's how we think it's happening. And again, we have a lot of data that seems to support this. In terms of other chromatin binding proteins, it seems to be the case that it just seems to be dependent on how long it's binding to DNA. So it's residence time, and whether that residence time is longer or shorter than the diffusion of formaldehyde. And so we've looked at other chromatin binding proteins like CTCF and a few others. And as long as it's stably bound to DNA, it gets cross-linked. But if it's not stably bound, then it's also evicted in the same way.
Nice talk, Dr. Teves. I have a question regarding the single molecule tracking that you showed in the early part of your talk. Usually the residence time would also depend on the cofactors and the motif to which the transcription factor is binding. So with respect to SOX2, when you switch domains, you said you're following it up with CHIP, but I'm curious to know if they bind to similar motifs, or it is, gene-specific context will,
that's exactly what we're trying to look at now. So SOX2, as you identified, usually, I mean, this is pretty common for a lot of transcription factors, for them to bind cooperatively or in conjunction with another transcription factor. SOX2 is the same way. It usually binds to OCT4, or it binds with OCT4 to DNA binding sites. But that's exactly the question we're trying to answer now. And in fact, it's now getting sequenced. So we don't know the answer yet, but we're hoping to know soon, and that hopefully will tie in pretty well with the single molecule imaging.
Okay. We have a question from our YouTube audience where they would want to know, Bhaskar Maji wants to know if they can control cancer by this mechanism.
By mitotic bookmarking, I'm assuming that's what they mean. That's a great question. I think so. I mean, I don't see why it wouldn't be, but I think it would have to identify what are the key main transcription factors that are involved in the driving the cancer state, and then asking whether they're important for this type of memory. And perhaps changing that transcription factor behavior would then affect cancer biology. That's an interesting idea. We haven't explored it, but I think it's a good idea.
We have two questions from Ashish. Maybe we can unmute Ashish and he can ask the questions. Ashish, you're unmuted, so you can ask the question.
Oh, yeah, okay. So first question is since we are saying that TBP depletion is affecting RNA pol3 transcription, so do you see the effect of this depletion in your nascent RNA sequencing?
Yeah, we do actually. It's a little bit less pronounced. So here the net-seq is on the bottom here in blue. You can see that it is depleted when you deplete TDP, but the net-seq, the problem with net-seq is that it is contaminated by mature RNAs, and one of the biggest source of contamination is actually tRNAs and ribosomal RNAs. And so part of this signal, part of the remaining signal might still be this contamination, but even then we still see this reduction by net-seq.
Okay, and this does this?
So yeah, we think definitely that pol3 is affected.
Okay. Then another thing I wanted to ask was this in interface chromatin also, is this exclusion of TF could be happening, and like you would be getting these things off your chromatin because of the fixation?
that is a great question. We can really only measure this by imaging. And when you image interface cells with or without fixation, they look the same because they're just nuclear, they're nuclear signal. But that said, if I can speculate, I would speculate that it is still happening in interface. It's just that we can't see it by imaging. But probably what this means is that things that are dynamically bound are less cross-linked than the ones that are stably bound. And so, things like ChIP-seq, for example, will still measure the transcription factors that are really stably bound, but you may lose the ones that are more dynamic. So the signal might be lost in those ones.
So what do you think could be the solution to this, like to get data that is more representative of the actual state?
Maybe,
yeah, so we're trying the cut and tag for that.
Yeah, maybe comparing chip with cut and tag will tell you what are the more dynamic binding sites which are getting excluded from the chip signal.
Right.
Yeah, that's exactly what we're trying to do. But it's really hard to know what you're missing if it is really dynamic binding. Like, what does that mean in terms of its actual function? It's unclear to me whether, whether that type of binding is more related to noise or related to its function of scanning the genome until it actually finds its target site, in which case is it something you really care about? Unclear. For me, by single molecule imaging, I can measure those under live conditions and I can know what they are, and I don't need necessarily like genome level locus resolution to do that type of assay. But yeah, I get this question quite a lot, and it's because I think it scares everybody. But whether you care about those dynamic binding is I guess the more important question.
Is there time? Could I ask another question?
Okay, ask away.
Yeah, so about this TBP binding, although it seems since if you knock out TBP, it's lethal, so it must be very important. So have you looked at or have you considered that single cell methods, that it may be very heterogeneous in its action?
Yeah, that's a great point, and especially since the level of depletion that we see is an aggregate level, and we don't necessarily know, at a single cell level whether it is 90% at all cells or a variation around that. It's possible that we can get more information by single cell analyses, but I mean, we expected global shutdown and we don't see that. And so already I feel like that is a big enough result for us that we don't necessarily need to know the level of variability at the single cell level. But, maybe reviewers might disagree with me on that. We'll see.
Thank you.
Also, great effort in chasing the TBP story because it feels like a marathon.
It's an unending marathon, unfortunately. I do see a hand up.
Yes.
Loda?
Yeah. Can you hear me? Can you hear me?
Yep.
Okay. So the question is that when you do these domain swap experiments in SOX2, is there any consequence for the cells?
So at this point, we are doing them in an overexpression system. So we're not affecting their endogenous proteins, which is important because SOX2 is an important transcription factor for ES cells. And so we're hopefully isolating the essential function from the modulation that we do with the transcription factors.
Okay. And in your TVP story, you are telling you are depleting TVP, but the question is that whether you are depleting all or the endogenous one is still there.
Oh, this is all the endogenous. So these are gene edited, CRISPR-Cas9 gene edited. So all endogenous proteins have the
Okay.
AID tag. So they're all depleted.
So if it is lethal, then how do you measure things actually?
So they are still alive by 6 hours. They actually start to die by 24 hours. By 24 hours, we get about 30 to 50% cells alive, remaining alive, and then it precipitates quite a bit after that. So at the time point that we're looking at, they're still alive.
Okay.
And so, and they're still transcribing RNA, right? So.
Okay. And one
except
question
for
about
Pol3.
Okay. One question about your formaldehyde experiments. So you are arguing that this is a artifact of formaldehyde. One possibility is that formaldehyde is just quenching the fluorescence. Anyway, the fluorescence will not come after formaldehyde treatment. So it's not that the protein is not there. It's that the fluorescence is not there. So there are two different things.
Yep, that's a great question. We tested this by switching fluorophores. So we switched tags so generally GFP is affected by formaldehyde, whereas M-cherry is not. And so we tested that and we saw the same effect, meaning that we still see this eviction from mitotic chromosomes. And then we also switched fixation method. So as you mentioned, formaldehyde could quench fluorophores, but ethanol does not. And so we also tried this with ethanol and methanol methods of fixation, and we saw the same exact thing.
Okay.
So we tried a whole bunch of different ways to make sure that we're seeing what we think we're seeing.
Okay. Thank you very much.
Yep.
We have one last question from Annapurna, and she would like to know if the fixation problem have implications for chip-based experiments too, and the alternatives for it. I think it's the cut and pack.
Yeah, I mean, I think it would. I mean, I would speculate that it would, just based on what we see by imaging. But it probably would mostly affect the dynamic binding, whereas the specific or the long-lived binding is generally not affected. And there's some evidence for this in the literature. So historically, transcription factors like c-Myc and other transcription factors are notoriously difficult to chip. And then later on, by imaging, they found that these transcription factors that are really hard to chip are actually very dynamic binding. They have very short residence times on the DNA. And so you can imagine that when you cross-link these for these transcription factors, you're probably not catching the dynamic binding of these events, right? So that's one way that chip would be detrimental to measure DNA binding. And hopefully the alternatives are these native conditions of cut and tag and cut and run that was developed in the Hennekoff lab. There you don't use formaldehyde, and hopefully you're catching these dynamic behaviors.
We have another question from Priyanka. Maybe we can unmute Priyanka and she can ask the question. Priyanka, are you around? Okay, I'll read out the question. Is there any difference between the complex proteins associated with Pol2 versus Pol3? Could that factor is causing the change in transcription between TBP wild type and null state of Pol2 and Pol3 dependent transcription?
So for the first question, there is a difference in the TBP complex that's found in Pol2 versus Pol3. So I just showed you earlier that in Pol2, TBP is in complex with TF2D. It's a very big complex. Whereas in Pol3, TBP is in complex with what's called TF3B, so transcription factor 3B for RNA polymerase 3. And the binding partners in that complex are different proteins. We're unclear whether they're the ones that are differentiating whether TBP is needed or not. But it's something that we're trying to pursue right now. So unclear at this time, unfortunately, but hopefully we will have a follow-up project precisely on POL3 and POL1 transcription.
I see no more questions. Thanks a lot, Dr. Teves.
Thank you so much for the invitation. Hopefully that there's a lot of really good questions, and thank you everybody for your questions. It's actually the most questions I've had in a seminar, so it's great.
Yeah, thank you.
Yep, thanks you. Have a good day.